cell-to-cell signaling usually occurs via the use of some sort of signaling molecule, with the exception of the nexus junctions and connexons. These cells talk with one another using signaling molecules. A cell will release a molecule, and that molecule will bind to a receptor on or in a target cell, and then it will create some sort of cascade within the target cell for the target cell to elicit some sort of response. And these signaling molecules can be a different types of chemical compounds. So when we talk about signaling molecules, we typically break them up into four main groups. We talk about peptides. We talk about steroids. We talk about biogenic amines. And we talk about eicosanoids. And so we're going to talk about each one of these independently as they act a little bit differently. They're sort of synthesized a little bit differently. And so it's important to know just a little bit of information about their production and their action as we move forward. Now, we're not going to focus on the detailed chemistry and the chemical makeup of the individual compounds, but it is important to understand their generic makeup. So first is we are going to start with our peptides, okay? And so if we think about our peptides, right, are just combinations of amino acids, okay? And so these amino acids are put together in sort of the rough ER. So you have your start in the rough endoplasmic reticulum, right? And with that, in the rough endoplasmic reticulum, you have basically a signaling group that tells the rough endoplasmic reticulum to start synthesizing this group of amino acids. And so as the amino acids are added, you end up with something called a pre-pro hormone. Okay? And your pre-pro hormone is the amino acid string plus this sort of signaling molecule to tell the rough endoplasmic reticulum to make this sort of structure. Now, inside of the rough endoplasmic reticulum, there are some peptidases that will cleave off that sort of start portion, and you are left with what's called a prohormone that will be shipped off to the Golgi apparatus for packaging. So this is going to get shipped off to the Golgi. Okay, now inside of that Golgi, right, is the prohormone's going to get packaged, and with that are going to be packaged to some enzymes along with it. And so that, or not hormones, those enzymes will start to cleave off a portion of that prohormone, and it will leave you with the active hormone. Okay, now whether this cleavage of the part of the peptide happens before or after that vesicle leaves the Golgi is sort of debated, and it can be specific to the individual molecule, right? But the Golgi is going to package that and ship it off to the outside of the cell via exocytosis. And what's going to happen is that hormone, right, will eventually end up in the bloodstream in its active form so that it can get taken off to its target tissue. A good example of a peptide hormone that follows this sort of cascade is the hormone known as vasopressin, or in humans, they typically refer to this as ADH, or antidiuretic hormone, right? And so what happens is, if you look at the production of vasopressin, is when it starts off in the rough endoplasmic reticulum, you get a signal peptide, and that signal peptide is combined with this sort of like complex, which is pro-vasopressin plus pro-neurophysin plus a glycopeptide. Now, inside of the rough endoplasmic reticulum, that signal peptide is sort of cleaved off, and you end up with the pro-vasopressin, pro-neurophysin, and then your glycopeptide, and that is then shipped off to the Golgi complex. Right? So this is going to go to your Golgi. Right? And once that goes to the Golgi, it will get packaged with some endopeptidases, some more enzymes, and that will split that into your vasopressin, which is your active hormone, and then you have the proneurophysin with the glycopeptide, which will further get split later on. Right? But notice, right, you start with this sort of complex here, your signal peptide plus your prohormones, right? And this is your pre-prohormone, right? Then it cleaves off that signaling peptide, and you're left with your prohormone. And then that sort of is cleaved again to get you to your active form, right? Which is your hormone. So that's how peptide messengers are sort of put together. Now, peptide messengers will go and they will bind to receptors on cell membranes, and we'll talk about how that works a little bit later. The next type of signaling molecule is the steroid hormones. And steroid hormones are a little bit different because they are lipophilic, right? They are fat attracting or lipid attracting, right? So they are not hydrophilic like the other signaling molecules. And there are two major groups of steroids that we talk about. There are the adrenocorticosteroids, which can further be broken into your mineralocorticoids and your glucocorticoids. And your mineralocorticoids regulate minerals inside of the body. Your glucocorticoids are known as your stress hormones. And then your second major group are your reproductive hormones, or what we refer to as sex hormones. Okay. Now, all of your steroid hormones are derived from cholesterol. Okay. They are derived from cholesterol. Now, cholesterol is synthesized, and the steroids, right, are derived from this cholesterol. And what makes this a little bit unique is because of their lipophilic nature, steroid hormones are not stored. Okay, so your other signaling molecules can be sort of synthesized and then stored in a cell until time of release. Your steroid hormones work a little bit different in that they are released at the time of synthesis. So if we want to upregulate the presence of steroids and their activity, we have to upregulate the synthesis. Okay, so that's a little bit different than some of your other signaling molecules where we can upregulate like the release of them. Okay, so your steroids, they are not stored. Now, because your steroids, right, they are lipophilic. All right. Now, because of that, they will basically cross the cell membrane. And so you will have your receptor inside of the cell, like so. So here's our signaling molecule, and it will bind to a receptor inside of the cell. So it will pass through that cell membrane and it will bind to the receptor on the inside of the cell. That is different than how your peptide messengers worked, where the peptide messengers had receptors on the cell membranes. These have receptors inside of the cell membrane, and they often serve as transcription factors, which upregulate the rate of transcription. The next type of signaling molecule is a biogenic amine. And from its name, I'm sure you can guess that biogenic amines have an amine group with them. Right? So they have an amine group associated with them. All right? And these are synthesized from amino acids. So these are basically derivatives of amino acids. And some of the most famous biogenic amines, right, are things known as catecholamines. And your catecholamines are derivatives of tyrosine. And these are things that you've probably heard of, like dopamine or epinephrine. Okay, so these often act as neurotransmitters, paracrines, and in some instances, hormones. Okay, but they are all derived from amino acids. Okay, and they will have receptors on the cell membrane. So they won't pass through the cell membrane like steroids do. They'll have receptors on cell membrane. So we can put that down here. Receptor on cell membrane. Okay. And then your last signaling molecule are your eicosanoids. Your eicosanoids are derived from something known as arachidonic acid. And arachidonic acid is a polyunsaturated fatty acid. Okay, and so these are sort of derivatives of a lipid, right? And they undergo some conformational changes at which they are promoted for cell signaling. Now, despite the fact that they are derivatives of the fatty acids, they cannot be transmitted across the cell membrane, and they use some sort of transporter to get across the cell membrane, right? And these ones you've probably heard of, things like prostaglandins, which are involved in the inflammation process or associated with pain. Okay, and so these are some of the ones that we talk a little bit more when we talk about the immune system, right? And we talk about the inflammatory pathways. So we'll see those a little bit later on as well. So remember, right, your four classes or four different types of signaling molecules. You have your peptide messengers, you have your steroids, you have your biogenic amines, and you have your eicosanoids, each a little bit different in their synthesis process each a little bit different in how they're transported, and each a little bit different in how they might interact with a target cell. So those are kind of important things to remember as we move forward, and we talk about them a bit later.